Hello students, welcome to Read Med Prep Academy channel. Today in Principles and Processes of Biotechnology Part 2, we shall discuss about the advancements in modern biotechnology. Modern biotechnology embraces all the genetic manipulations, protoplasmic fusion techniques and the improvements made in the old biotechnological processes. Some of the modern biotechnology advancements are genetic engineering and recombinant DNA technology. Genetic engineering or recombinant DNA technology or gene cloning is a collective term that includes different experimental protocols resulting in the modification and transfer of DNA from one organism to another. Conventional recombination involves exchange or recombination of genes between homologous chromosomes during meiosis. Recombination carried out artificially using modern technology is called recombinant DNA technology or DNA technology. It is also known as gene manipulation technique. This technique involves the transfer of DNA coding for a specific gene from one organism to another organism using specific agents like vectors or using instruments like electroporation, gene gun, liposome mediated, chemical mediated transfers and microinjection. Now, what are the steps involved in recombinant DNA technology? First is isolation of the DNA fragment containing a gene of interest that needs to be cloned. This gene of DNA is called insert. Next is PCR, polymerase chain reaction. Is a common laboratory technique used to make copies, millions of a particular region of DNA. What is vector? Generation of recombinant DNA or DNA molecule by insertion of the DNA fragment into a carrier molecule. The carrier molecule is called a vector that can self-replicate within the host. Here you can see in the image a DNA fragment which is to be impregnated called the insert, and a vector which is a carrier DNA molecule. When the both are combined, you get a recombinant DNA. Selection of the transformed host cells is carrying the rDNA and allowing them to multiply, thereby multiplying the rDNA molecule. The entire process thus generates either a large amount of rDNA or a large amount of protein expressed by the insert. Wherever the vectors are not involved, the desired gene is multiplied by polymerase chain reaction technique. The multiple copies are injected into the host cell protoplast or it is shot into the host cell protoplast by a shotgun method. Here you can see a shotgun which has a pulse of helium, a cartridge holder having a gun barrel and the microprojectiles which are gold or tungsten particles coated with the recombinant DNA molecules. When the gun is used, the particles are impregnated or injected into the plant protoplast. This is the gene gun method. So what are the steps involved in the recombinant DNA technology? To sum up, the bacterium is taken which has a nucleoid containing the bacterial chromosome and a plasmid. The plasmid is taken out and it is cut at certain places, and a DNA of the chromosome is taken of interest and the genes of interest are removed from it. So the isolation of the plasmid DNA and the DNA containing gene of interest is taken, and the gene of interest is inserted into the plasmid, and the plasmid is put into the bacterial cell and it forms a recombinant bacterium. And the cells cloned with the gene of interest and identification of the desired clone, and the copies of genes are made within the bacterial cell, and the various applications are used, like the gene is used to alter bacteria for cleaning up toxic waste or for basic research purposes or introducing into the plants for the better products and better plants. Now, what are the tools for genetic engineering? In order to generate recombinant DNA molecule, certain basic tools are necessary. The basic tools are enzymes, vectors and host organisms. The most important enzymes required for genetic engineering are restriction enzymes, DNA ligase and alkaline phosphatase. Let us discuss one by one. The restriction enzymes. The two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated in the year 1963. One was the enzyme which added methyl groups to the DNA, while the other cut the DNA. So, based on their mode of action, restriction enzymes are classified into restriction exonucleases and restriction endonucleases. Restriction exonucleases are enzymes which remove the nucleotides one at a time from the end of a DNA molecule. Example, BAL31 exonuclease 3. Restriction endonucleases are enzymes that cut the DNA and are called restriction endonucleases, which can be done at certain places within the DNA molecule. A restriction enzyme or restriction endonuclease is an enzyme that cleaves the DNA into fragments at specific recognition sites within the molecule known as restriction sites. The endonucleases are enzymes which break the internal phosphodiester bonds within a DNA molecule. Example, HIND2, ECOR1, PVUL, BAMH1, TAQ1. To sum up the activity of nucleases, the endonucleases causes internal cuts of the DNA molecule. Exonucleases, nucleotides are removed from the ends of the DNA molecule. So, restriction endonucleases cleave the DNA at the restriction sites within the DNA molecule and break the DNA molecule into fragments. The restriction enzymes are called molecular scissors. These act as foundation of recombinant DNA technology. These exist in many bacteria where they function as a part of the defense mechanism called restriction modification system. There are three main classes of restriction endonucleases: type 1, type 2, type 3, which differ slightly by their mode of action. Only type 2 enzyme is preferred for use in recombinant DNA technology, as they recognize and cut the DNA within a specific sequence, typically consisting of 4 to 8 base pairs BP. Now, you can see in this table type 2 restriction enzyme with source, recognition and cleavage unit. In this table, the restriction enzyme ALU1, the microbial source is Arthrobacter luteus. The recognition sequence 5-AGCT3-. Another strand 3-TC GA5-, and the cut is made 5-AG and CT3-. In the complementary strand, the cut is made between C and G. So, the fragments obtained AGTC, CTGA, which has blunt ends. The restriction enzyme BAMH1 is taken from Bacillus amyloliquefaciens, which has 5-G, GATCC3-, and the complementary strand 3-CCTAG, G5-. And the cut is made between G and G in the above strand and the complementary strand between G and G. So, the fragments obtained is G, CCTAG, and another fragment GATCC and G. These are sticky ends. In the restriction enzyme, ECOR1 is obtained from Escherichia coli. The above strand 5-G, AATTC3-, and the complementary strand 3-CCTAG, G5-. You can see the cut between G and A in the above strand and the complementary strand between G and G. So, the fragments obtained is G, CTTAA, and another fragment AATTC and G. These are sticky ends. HAE3, which is a restriction enzyme obtained from Haemophilus aegypticus. The 5-3- strand 5-GG-CC3-. The complementary strand 3-CC-GG5-. and the cut is done between G and C in the above strand and the complementary strand between C and G. So the fragment obtained is GGCC and CCGG, which have blunt ends. The restriction enzyme Hind 3 obtained from Haemophilus influenza, the above strand 5-A AGCTT3- and the complementary strand 3-TT CGA A5-. And the split is made between A and A in the above strand and the complementary strand between A and A. And the fragments obtained ATTCGA, AGCTT and A, and these have sticky ends. So the restriction enzyme Hind 2 always cuts the DNA molecules at a point of recognizing a specific sequence of 6 base pairs. This sequence is known as recognition sequence. Today, more than 900 restriction enzymes have been isolated from over 230 strains of bacteria with different recognition sequences. This sequence is referred as restriction site and is generally palindromic, which means that the sequence in both DNA strands at this site reads same in 5' to 3' direction and in the 3' to 5' direction. Example, Malayalam. This phrase is read the same in either of the direction. The palindromic repeats a symmetrical repeated sequence in DNA strands. 5' CATTATATATG 3'. Repeated in the opposite direction, 3' GTAATATATTAC 5'. Here you can note that the sequence of the base pairs in the reverse direction when compared to the first sequence. Restriction endonucleases are named by a standard procedure. The first letter of the enzymes indicates the genus name, followed by the first 2 letters of the species. Then comes the strain of the organism, and finally a Roman numeral indicating the order of the discovery. For example, when you take ECO capital R1 is Escherichia, represented by E. coli, represented by CO. The strain capital R capital Y 13 is represented as R. And since the enzyme was the first endonuclease to be isolated, represented as 1 to be discovered. Now, what are symmetric cuts? The exact kind of cleavage produced by a restriction enzyme is important in the design of a gene cloning experiment. Some cleave both the strands of DNA through the center, resulting in blunt and flush end. These are known as symmetric cuts. What are asymmetric cuts? Some enzymes cut in a way producing protruding and recessive ends known as sticky or cohesive ends. Such cut is called staggered or asymmetric cuts. We have seen the examples in the table. Here you can see, to show once again, the sticky and blunt ends. The sticky ends 5' GAATTC 3' is one strand, and the complementary strand 3' CTTAAG 5'. When the enzyme ECOR1 is used to digest, the break occurs between G and A in the above strand and between A and G in the complementary strand. So this results in the formation of 2 fragments, G in the above strand and CTTA in the lower strand as one fragment, and AATTC in the above fragment and G in the lower part of the complementary strand, forming 2 fragments which have sticky ends. In the lower image you can see 5' CCCGGG 3', and the complementary strand 3' GGGCCC 5'. When SMAL enzyme is used to digest, the break occurs between C and G in the above strand and the complementary strand between G and C. So it results in blunt ends. One fragment having a CCC in the above strand and GGG in the complementary strand, and another fragment having GGG in the above strand and CCC in the complementary strand. Now let us move on to DNA ligase. This enzyme joins the sugar and the phosphate molecules of double-stranded DNA, dsDNA, with 5' PO4 and a 3' hydroxyl in an adenosine triphosphate dependent reaction. This is isolated from T4 phage. Here you can see the DNA strand with a hydroxyl group and another DNA strand with a phosphate group, and the hydroxyl and the phosphate group are joined by the enzyme DNA ligase, and ATP is reduced to AMP and PPI gives 2PI, and the two strands are joined together and a new 3'5' phosphodiester bond is formed. Now something about the alkaline phosphatase. It's a DNA modifying enzyme and adds or removes specific phosphate group at 5' terminus of double-stranded DNA, dsDNA, or single-stranded DNA, ssDNA, or RNA. Thus, it prevents self-ligation. This enzyme is purified from bacteria and calf intestine. Here you can see the plasmid when it is cut open, forming a linear DNA. The DNA ligase is used in attaching the hydroxyl and the phosphate group. Whereas the alkaline phosphatase, when it is used, it has no reaction. The DNA ligase has no reaction, but the alkaline phosphatase helps in impregnating the insert with the vector. So HO to P and P to HO, the foreign DNA fragment is attached to the vector in the presence of DNA ligase after the activity of the alkaline phosphatase. So what are the steps in the formation of the recombinant DNA? The plasmid and the foreign DNA are cut by the same restriction enzyme, in this case ECOR1, and annealing occurs, that is the joining of the foreign DNA gene with the vector DNA, resulting in the formation of a new recombinant molecule. When the new recombinant DNA molecule is formed, the sealing occurs. The nicks between the plasmid and the insert are nicked or sealed by the DNA ligase. Now, what are vectors? Another major component of a gene cloning experiment is a vector such as plasmid. A vector is a small DNA molecule capable of self-replication and is used as a carrier and transporter of the DNA fragment, which is inserted into it for cloning experiments. What are the types of vectors? Cloning vector and expression vector. So cloning vector is used for cloning of DNA insert inside the suitable host cell. Expression vector is used to express the DNA insert for producing specific protein inside the host. Now, what are the properties of vectors? Vectors are able to replicate autonomously to produce multiple copies of them along with their DNA insert in the host cell. It should be small in size and of the lower molecular weight, less than 10 kb, kilobase pairs in size, so that the entry and transfer in the host cell is easy. Vector must contain an origin of replication so that it can independently replicate within the host. It should contain a suitable marker such as an antibiotic resistance to permit its detection in transformed host cell. Vector should have unique target sites for integration with the DNA insert and should have the ability to integrate with the DNA insert it carries into the genome of the host cell. Most of the commonly used cloning vectors have more than one restriction site. These are multiple cloning sites called MCS or polylinker. Presence of MCS facilitates the use of restriction enzyme of choice. Here you can see the properties of vector that has been summed up. So unique restriction enzyme cleavage sites have been shown of PST1, BAMH1, SAL1, ECOR1, HIND3. ORI is an origin of site of replication, and you have a part called the selectable marker. A cloning vector must contain an origin of replication, otherwise called ORI, recognized in the host cell, so that it is replicated along with the DNA that it carries. It should carry a selectable marker traits that enable the cells containing the vector to be selected or identified. A cloning vector needs a single cleavage site for each of one or more restriction enzyme used. So what are the features required to facilitate the cloning into a vector? The origin of replication or ORI. This is a sequence from where replication starts, and piece of DNA when linked to this sequence can be made to replicate within the host cells. Selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating the non-transformants and selectively permitting the growth of the transformants. Cloning sites. In order to link the allele DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. What are the types of vectors? Plasmid, PBr322 plasmid, Ti plasmid bacteria are some of the examples. Now let us talk about plasmid. Plasmids are extra-chromosomal, self-replicating, double-stranded circular DNA molecules found in the bacterial cells in addition to the bacterial chromosome. Plasmids contain genetic information for their own replication. Here in this image you can see the bacteria having the bacterial chromosome in the nucleoid, which is circular in shape, and the extranuclear chromosome, that is called the plasmids, which are circular in shape. PBr322 plasmid is a reconstructed plasmid and most widely used as a cloning vector. It contains 4361 base pairs. In PBr, small p denotes plasmid. B and R, which are capital respectively, the names of scientists Bolivar and Rodericks who developed this plasmid. The number 322 is the number of the plasmid developed from the laboratory. It contains AMP to the power of R and TET to the power of R, two different antibiotic resistant genes, and recognition sites for several restriction enzymes. HIND3, ECOR1, BAM1, SAL1, PVU2, PST1, CLAV1, ORI and antibiotic resistant genes. ROP codes for proteins involved in the replication of the plasmid. Here you can see in this image the different sites where the enzymes work. ECOR1, HIND3, BAMH1, and the ampicillin resistance genes AMP capital R to the power, AMP to the power R, TET to the power R, ORI, ROP, PVU2. So this totally constitutes the PBr322. Now Ti plasmid bacteria. Ti plasmid is found in Agrobacterium tumefaciens, a bacteria responsible for inducing tumors in several dicot plants. The plasmid carries transfer TRA gene, which helps to transfer tDNA from one bacterium to another bacterium or plant cell. It has ONC gene for oncogenicity, ORI gene for origin for replication, and INC gene for incompatibility. tDNA of Ti plasmid is stably integrated with the plant DNA. Agrobacterium plasmids have been used for introduction of genes of desirable traits into the plants. Here in this image you can see the different genes that produce auxin, cytokinin, opine catabolism gene, origin of replication ORI, and virulence region. So the entire plasmid is the Ti gene. The competent host. The propagation of the recombinant DNA molecules must occur inside a living system or host. Many types of host cells are available for gene cloning, which includes E. coli, yeast, animal or plant cells. Here you can see the different host cell types. The prokaryotic host, bacteria like E. coli, Bacillus species, Pseudomonas species, Streptomyces. The eukaryotic host, yeast like Saccharomyces, algae like Chlamydomonas, fungi like Aspergillus and Neurospora. The type of the host cell depends upon the cloning experiment. E. coli, Escherichia coli, is the most widely used organism as its genetic makeup has been extensively studied. It is easy to handle and grow and can accept a range of vectors and has also been studied for safety. It has nucleoid which has the bacterial chromosome and it has plasmid. One more important feature of E. coli is to be preferred as a host cell is that under optimal growing conditions, the cells divide every 20 minutes. Since the DNA is a hydrophilic molecule, it cannot pass through the cell membranes in order to force bacteria to take up the plasmid. The bacterial cells must first be made competent to take up DNA. Here you can see a bacteria with the bacterial DNA and the plasmid undergoes replication. When it replicates, the bacterial chromosome also duplicates along with the plasmids. Two cells are formed identically. When a bacterial cell is impregnated with the plasmid into the nucleoid chromosome, when it undergoes replication, the similar cells are formed with the plasmid impregnated bacterial chromosome. This is done by treating them with a specific concentration of a divalent cation such as calcium. So the competency of the E. coli cell is increased. The recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees centigrade heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. On the left side in the image you can see a non-competent E. coli. E. coli that has not been made competent. That means it will not take up the plasmid introduced into the environment. Whereas on the right side you can see the E. coli is made competent. How? By treating them with calcium chloride or by heat shock or through electroporation. This gives better membrane permeability and forms spores, enabling the plasmid uptake. The bacteria E. coli is used as the host cell. If E. coli and the recombinant plasmids are mixed together in a test tube, the recombinant plasmid enters very easily into the cell. Thus how the recombinant DNA is introduced, or it can also be done by electroporation or other methods which I have already mentioned earlier. Now here you can see, to sum up, the donor cells are taken, the foreign DNA is removed, treated with the restriction enzymes and the fragments are made, a bacterial cell E. coli is taken and a plasmid is removed which acts as the vector DNA, and a portion of the plasmid DNA is cut and the ligase enzyme is used which helps in the impregnation of the foreign DNA along with the plasmid, and the recombinant DNA is introduced into the E. coli cell, and this helps in further development and multiplication of the clones of the recombinant DNA, and the clones of engineered bacteria are produced in a medium and the recombinant product is obtained, and downstreaming is done where the separation and purification of the product is done. For the expression of the eukaryotic proteins, eukaryotic cells are preferred because to produce a functionally active protein, it should fold properly and post-translational modification should also occur, which is not possible by prokaryotic cell like the E. coli. So today in Principles and Processes of Biotechnology Part 2, we discuss about the advancements in modern biotechnology like the genetic engineering or the recombinant biotechnology. Thank you. Kindly subscribe, like, share and comment to channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.